right now, in every single green leaf right outside your window, the most important chemical reaction on Earth is just actively failing. Like 25% of the time, it fails because of this billion-year-old blind spot.
I mean, it is a staggering level of inefficiency. And it's happening inside the very engine that, drives the creation of the oxygen we breathe.
Huh.
And really, the base of the entire global food chain.
Right. Welcome, learner, to today's deep dive. We are looking at a comprehensive breakdown of photosynthesis. Because, when you walk into an elementary school, you always see that one classic science poster.
Oh, yeah, the cartoon one.
Exactly. The happy, cartoonish green flower. You've got a bright yellow sun with, sunglasses shining down, a blue watering can pouring on the roots, and a big arrow pointing to the leaf that just says food.
Right. Light plus water equals plant grows.
Yeah, and we accept that when we're 7. But when you pull out the microscope and look at the biological reality we are diving into today, that cartoon just dissolves. It turns into this wildly complex, hyper-industrialized biochemical factory that is constantly on the brink of making massive, costly mistakes.
It really is. Today is all about moving past that grade school poster. We're looking at the foundational mechanics of photosynthesis. Not just what it is, but the actual atomic machinery that makes it happen.
Right. The real nuts and bolts.
Exactly. And, the evolutionary quirks that left it so surprisingly flawed.
Okay, let's unpack this, because before we can even begin to understand how plants manufacture their own food out of thin air, we really have to look at the currency of energy driving the whole factory.
Yeah, you have to understand the power source first.
Right. And I've always heard ATP described as a rechargeable battery, but looking at the mechanics of it in the source material, that feels a bit, I don't know, passive. It seems more like a tightly compressed spring.
Oh, a compressed spring is a much better way to visualize it. I mean, ATP adenosine triphosphate, it stores energy in the chemical bond of its third phosphate group.
Okay.
So when the cell needs to do work, an enzyme called ATPase facilitates hydrolysis. That basically means using water to break that specific bond.
And it doesn't just slowly drain power like a battery, right?
No, not at all. It snaps. Breaking that bond releases a highly specific burst of energy. It's about 34 kilojoules per mole.
Wow, 34 kilojoules. And that literal physical snap, like that release of tension, is what forces other molecules to change shape or move across a membrane.
Exactly. It's a mechanical transfer of power. That 34 kilojoule burst drives virtually everything. Active transport of nutrients into the roots, building up large molecules, replicating DNA.
It does all of that.
Yeah. And once that spring is sprung, you are left with ADP, adenosine diphosphate, and a loose inorganic phosphate.
Right, the uncoiled spring. But the cell can just use energy from other reactions to force that phosphate back on, right? Like specifically redox reactions where electrons are passed down a chain.
Correct. It basically just compresses the spring again. And what's fascinating here is the sheer universality of this mechanism.
What do you mean?
Well, this specific molecule, this exact chemical spring, is highly conserved across all domains of life. It's not just a plant thing. The ATP in a blade of grass is identical to the ATP powering the neurons in your brain right now as you listen to this deep dive.
That's incredible to think about.
It really is. If anything interrupts the synthesis or hydrolysis of ATP, the organism dies almost instantly.
Okay, so we have our universal mechanical spring. We have the energy currency. I guess the next logical step is understanding the specialized solar hardware plants use to capture sunlight to actually, compress those microscopic springs.
Right, the solar panels.
Yeah. Here's where it gets really interesting. When we think of leaves, we just think green. But they aren't just one flat color, are they?
No, not at all.
They contain a whole cocktail of pigments. Chlorophyll A, chlorophyll B, carotenoids. Why all the variations? Like, if evolution is so ruthlessly efficient, why not just develop one perfect, all-absorbing black pigment?
Well, it really comes down to the physics of light and molecular resonance. Visible light is a spectrum of different wavelengths, right? And each carries a different amount of energy.
Yeah.
A pigment molecule has a very specific structural geometry. When light hits it, only certain wavelengths resonate perfectly enough with that structure to excite its electrons.
So it's like a tuning fork.
Exactly. Chlorophyll A, for instance, absorbs heavily in the red and blue wavelengths. But the physical structure of the molecule simply rejects green light, so it just reflects it back to our eyes.
Which means if a plant only relied on chlorophyll A, it would be throwing away a massive portion of the solar energy, bombarding it every second.
Yeah, they would be leaving an immense amount of power on the table.
Mm.
So by maintaining a cocktail of secondary pigments like the yellow-green chlorophyll B or the orange and yellow carotenoids, the plant broadens its net.
Ah, I see.
Each pigment absorbs a slightly different band of the spectrum and then transfers that captured resonance back to the primary chlorophyll. This combined absorption capability is what we call the action spectrum.
There was this wild experiment back in the 1880s by a scientist named T.W. Engelmann that proved this. And it just sounds like something out of a Victorian science fiction novel.
The Engelmann experiment, it's brilliant.
He essentially mapped this microscopic action spectrum using living creatures,
I
right?
think he did.
Yeah.
His setup was brilliantly elegant, especially considering they didn't have electronic sensors back then. He took a single microscopic strand of filamentous algae and just placed it on a slide.
Oh, okay.
Then he used a glass prism to split a beam of white light into a tiny rainbow. So he's projecting different colors across different sections of that single algae strand.
And because photosynthesis produces oxygen as a waste product, he needed a way to see which color was producing the most oxygen.
Right.
So he introduced oxygen-seeking bacteria into the water on the slide.
Yeah, he essentially used the bacteria as living data points. He just watched through his microscope to see where they swarmed.
And what happened?
They clustered densely around the sections of the algae bathed in blue and red light, and almost completely avoided the sections in the green light. He visually proved that the rate of photosynthesis perfectly correlates with the specific light absorption properties of those distinct plant pigments.
Wow. Okay, so we have the universal energy spring in ATP, and we have the multi-pigment solar panels casting a wide net for different wavelengths.
Yep.
How does the factory actually use this to build a plant? It's funny to me that older textbooks used to divide this assembly line into light reactions and dark reactions.
Yes.
That phrasing completely implies plants are doing half their work at night, which obviously isn't true if the whole system relies on the temporary products of the sunlight.
Yeah, you're touching on a historical naming flaw that confuses a lot of biology students. The entire process happens continuously in the daylight.
Right. So let's break down the actual stages.
Okay, so the first stage, the light-dependent stage, takes place on the thylakoid membranes inside the chloroplast. This is where photons literally, physically strike the pigment molecules.
And that excites them.
Very much so. It excites their electrons so violently that the electrons are ejected from the molecule entirely.
So the photon essentially knocks the electron right out of its orbit. But if the chlorophyll keeps losing electrons to this transport chain, doesn't it run out of ammunition? Like,
Right.
wouldn't the system just stall?
It would, yeah. Then there was where water comes in, and where plants perform one of the most violent chemical feats in nature. It's called photolysis.
Photolysis, meaning breaking with light.
Exactly. The plant uses the energy harvested from light to literally rip water molecules apart. It strips the hydrogen atoms to steal their electrons and uses them to replenish the chlorophyll.
And it just throws the oxygen away.
Just tosses it out.
I mean, the oxygen gas that every animal on Earth relies on to survive is literally just industrial exhaust from the plant's perspective. It only wanted the electrons from the water.
It is entirely a waste product to them. The true goals of this light-dependent stage are twofold. First, to compress that ATP spring we discussed, and second, to load the stolen hydrogen ions onto a carrier molecule called NADP.
Okay, so ATP and reduced NADP.
Right. Those two products are then immediately fed into the second stage.
The light-independent stage, the Calvin cycle.
Yeah.
And this is happening in the fluid matrix surrounding those solar panels, the stroma. This is where the plant finally pulls carbon dioxide out of the air to build something solid.
Exactly. The plant uses an enzyme, famously known as rubisco, to grab carbon dioxide gas and merge it with a 5-carbon compound already present in the plant called RoBP.
Okay, so it combines them.
Yeah. And through a cycle of chemical reductions, which are powered by snapping those ATP springs and using the hydrogen from the NADP, the plant eventually produces a crucial 3-carbon sugar called GALP.
GLP, glyceraldehyde 3-phosphate. I always just assumed plants made basic glucose, but GLP is so much more profound than that.
Oh,
It's like the
good.
ultimate modular biochemical Lego brick.
That's a great way to put it. Glucose is just one possible outcome. GALP is really the foundational building block for the entire physical structure of the organism.
So what else can it make?
Well, the plant can stick GLP molecules together to form sucrose for transport or starch for long-term energy storage. It can weave them into complex cellulose fibers to build rigid cell walls.
Wow. And if the roots are pulling up nitrates and phosphates from the soil, the plant can snap those onto the GLP framework too, right?
Exactly. To manufacture amino acids for proteins, lipids for cell membranes, and even the nucleic acids required to build DNA. Every physical component of a giant redwood tree ultimately derives from this microscopic 3-carbon sugar.
It is a breathtakingly productive biochemical pathway. Like, it literally weaves the physical world out of thin air and sunlight.
It really does.
But this brings us back to the failure we mentioned at the start. Hidden right in the middle of this beautiful, elegant assembly line is that key enzyme, rubisco.
Yes, the flawed engine.
Right. Chemically speaking, it is terribly designed for the job it has to do today. So what does this all mean?
Well, rubisco is arguably the most important enzyme on the planet. It is the gateway for inorganic carbon to enter the biological food chain.
Okay.
And yet its active site, the part of the protein that physically grabs the carbon dioxide, is remarkably imprecise.
I picture the active site of rubisco like a keyhole on a door. It's supposed to only accept the specific key, which is carbon dioxide, but the keyhole was forged just a little too wide and the lock is a bit sloppy.
That's a perfect analogy.
Because the carbon-oxygen double bonds in CO2 look structurally very similar to the oxygen double bonds in oxygen gas. So oxygen acts like a lockpick. It fits into the keyhole just well enough to trigger the mechanism.
And that structural similarity creates a disastrous competition.
Right.
In our modern atmosphere, rubisco frequently acts as an oxygenase instead of a carboxylase.
Meaning it grabs oxygen instead of carbon.
Right. It grabs an oxygen molecule and jams it into the 5-carbon RoBP molecule. This triggers a process called photorespiration.
Which is the exact opposite of what the plant is trying to achieve.
Completely counterproductive.
Yeah.
Instead of making the useful GLP precursor, the enzyme produces a useless, toxic waste product called glycolate 2-phosphate.
Yikes. So what does the plant do with that?
To survive, the plant then has to expend precious energy and previously captured carbon just to clean up the mess and recycle that waste. Photorespiration burns energy, wastes water, and actually forces the plant to exhale carbon dioxide back into the air.
I saw a statistic in the source that practically made me wince. For human beings, an enzyme that prefers its target molecule 80 times more than the alternative sounds pretty good.
Yep.
And rubisco is attracted to CO2 about 80 times more than oxygen. But the modern atmosphere is only 0.04% carbon dioxide and 21% oxygen.
Yeah, the math is brutal.
There is a massive, overwhelming sea of lockpicks for every one actual key. It means the enzyme is grabbing the wrong molecule constantly, reducing the efficiency of the entire process by 25%.
You have a 25% loss in productivity occurring at the very foundation of the global food chain. It's wild.
Wait, if evolution is usually this ruthless optimization engine, why are we stuck with such a flawed system at the center of life? Like, why wouldn't plants have evolved a tighter keyhole by now?
Well, you're assuming evolution looks forward. It doesn't. It only responds to immediate environmental pressures. What's fascinating here is, if we look at deep geological time, we get the answer.
Okay, take us back.
When the distant ancestors of modern plants first evolved the rubisco enzyme billions of years ago, the Earth's atmosphere was radically different. It was incredibly rich in carbon dioxide, and there was virtually zero free oxygen.
The lockpick didn't exist yet.
Zero competition. In that primordial environment, photorespiration was physically impossible because there was simply no oxygen to mistakenly grab.
So it didn't matter if the lock was sloppy.
Not at all. There was absolutely no evolutionary selection pressure for rubisco to develop a highly specific, tight active site. It just grabbed whatever bumped into it, and it was always CO2.
But then, the ultimate irony. Photosynthesis was so spectacularly successful that over hundreds of millions of years, it pumped the atmosphere full of oxygen.
Yep.
The plants effectively polluted their own environment with their own exhaust, poisoning the efficiency of their central enzyme.
Exactly. And because rubisco is so deeply embedded in the fundamental chemistry of life, a radical redesign of the protein structure was incredibly difficult to achieve through random mutation.
So how do they survive?
To compensate for having such a slow, inefficient enzyme in a high-oxygen world, plants just brute force the problem by producing absurd quantities of it.
Just sheer volume.
Yeah. Rubisco makes up roughly 30% of the total protein in a typical leaf. It is widely considered the most abundant single protein on planet Earth. simply because it is so bad at its job that plants need mountains of it just to survive.
Okay, this raises an important question. Evolution might have stalled on redesigning the enzyme itself, but the stakes for humanity today are astronomical.
They really are.
Billions of people rely on staple crops like rice, wheat, and soybeans. These are all what biologists call C3 plants.
Right. Meaning they rely entirely on this primitive, highly inefficient Rubisco setup.
So how are we supposed to feed a growing global population using an engine that misfires a quarter of the time?
Well, that inefficiency is the critical bottleneck in global agriculture. But while evolution couldn't easily redesign the Rubisco protein, it did eventually find a fascinating anatomical workaround in a small percentage of plants.
Oh.
We call it C4 photosynthesis. And understanding this hack is really the holy grail of modern plant science.
Okay, so C4 plants didn't fix the keyhole. They just moved the door. They hacked their own physical leaf anatomy to protect the enzyme, right?
Exactly, they isolated it. C4 plants like corn and sugarcane, which evolved in hot, bright, dry environments where photorespiration is most severe, developed a specialized internal structure.
Okay.
They physically separate the initial carbon capture from the Calvin cycle.
So they put the Rubisco in a sealed-off bunker.
That is functionally what the bundle sheath cells are. In the outer mesophyll cells of a C4 leaf, the plant uses a completely different, highly efficient enzyme, one that is absolutely immune to oxygen.
Okay, and that grabs the carbon dioxide.
Yes, it fixes that carbon into a temporary 4-carbon transport molecule. That transport molecule is then actively pumped deep into the inner bundle sheath cells, where the Rubisco is housed.
So they are using biochemical pumps to force-feed carbon dioxide into the bunker.
Yep.
They artificially create an environment around the Rubisco where the CO2 concentration is so overwhelmingly high that the oxygen just doesn't stand a chance.
Exactly. The plant expends a little extra ATP to run those cellular pumps, but the energetic trade-off is massively in its favor.
Whoa.
By saturating the Rubisco with carbon dioxide, photorespiration is virtually eliminated.
Wow.
At temperatures over 20 degrees Celsius, C4 photosynthesis drastically outpaces the standard C3 process. These plants grow faster, yield more biomass, and crucially, they lose far less water to evaporation.
Why less water?
Because they don't have to keep their leaf pores wide open all day scavenging for choice amounts of CO2.
That makes total sense. And if we connect this to the bigger picture, this brings us to the cutting edge of global humanitarian science.
It does.
Plant geneticists and agricultural researchers, particularly major consortiums working out of the UK with global partners, are actively attempting the most ambitious biological replumbing in human history.
Yes. The C4 rice project.
They are trying to engineer C3 rice to incorporate these C4 properties.
It is an incredibly complex challenge because, you aren't just tweaking one gene. You're trying to convince a rice plant to fundamentally alter its cellular architecture.
Right, to build those specialized bundle sheath bunkers.
Yes, and to express the distinct enzymes in the correct locations.
But the payoff, if they succeed, a fully realized, turbocharged C4 variety of rice could theoretically increase crop yields by up to 50%.
50%. Think about the macroeconomics of a 50% yield increase.
It's massive.
You are producing half again as much food on the exact same plot of arable land, using the exact same amount of fertilizer, and likely requiring less irrigation water because of the improved water use efficiency.
It fundamentally changes the math of human survival. We are talking about drastically reducing global deaths tied to hunger and malnutrition.
Tens of thousands of lives saved annually.
But it goes beyond just pure calorie counts, right? For a subsistence farmer in a developing nation, growing a crop that is 50% more productive means they are no longer just scraping by to feed their own family.
Exactly. They transition from subsistence to surplus. They have excess grain to sell at market.
And that sudden influx of capital allows a family to pay for medical care, allows them to pay school fees, keeping children in education rather than pulling them out to work the fields.
It proves how understanding the microscopic interactions of an electron bouncing around a pigment molecule can directly scale up to solving massive global humanitarian crises.
The micro dictates the macro.
It really does.
It's just wild. Let's retrace the path we just took today. We started with the physical snap of a tightly coiled ATP spring, releasing its 34 kilojoules of power.
Right?
Then we watched the leaf deploy a wide net of pigments to catch different frequencies of starlight, using that energy to literally tear water molecules apart just to steal their electrons.
And we followed those electrons into the Calvin cycle, where the plant weaves inorganic carbon into GLP, the universal building block of the physical world.
And then we confronted the great paradox, Rubisco, the sloppy, ancient enzyme forged in a prehistoric world, continually grabbing the wrong molecule and wasting a quarter of the plant's hard-earned energy.
Which ultimately led us to the bundle sheath bunkers of C4 plants.
Right, and the modern scientist trying to hack a billion-year-old biological flaw to rewrite the future of global agriculture.
It forces you to look at a simple forest or a field of crops with an entirely different level of respect.
Absolutely. The next time you, the learner, look at a green leaf on a tree or sit down to a simple bowl of rice, I hope you realize you aren't looking at a passive cartoon poster.
Far from it.
You are looking at a hyper-complex, dynamically flawed, but undeniably miraculous piece of biochemical engineering that is actively sustaining all life on Earth.
And before we close, this raises an important question to mull over long after you finish listening.
What's that?
Well, we established that the Rubisco enzyme is so inefficient today because it originally evolved in a primordial Earth atmosphere that was suffocatingly thick with carbon dioxide.
Right.
Today, through rapid industrialization and human activity, we are pumping massive amounts of carbon dioxide back into our modern atmosphere.
Oh, wow. I see where you're going.
So how will the rapidly rising global CO2 levels of climate change paradoxically impact the efficiency of global plant life? Will the deeply flawed enzyme suddenly find itself operating back in the ancient, carbon-rich environment it was originally built for?
That is fascinating.
It's a profound puzzle, really, connecting the deepest history of cellular evolution directly to our immediate ecological future.